Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Simon Kohl, founder and CEO of Latent Labs, a frontier AI lab working to transform the expensive, labor-intensive, and high failure rate processes of drug discovery into automated drug design. Welcome, Simon.
Hi, Nicole. It's great to be on.
So today, Simon and I will be discussing Latent Labs' emergence from stealth, as well as AI protein design and drug discovery acceleration in more detail, and the importance of democratizing research. But before we sink our teeth into our central topic, Simon, perhaps you could briefly introduce yourself to listeners, describe your journey to doing what you do today with Latent Labs, and the passions that drove you, so to speak. I mean, I believe you previously worked at Google DeepMind and indeed played a pivotal role in the development of AlphaFold.
Yeah, absolutely. Let's start there. That sounds great. So, my name's Simon Kohl. I'm the founder and CEO of Latent Labs. As you just said, my career really after my PhD in Heidelberg, Germany, started at Google DeepMind in London. I pretty early on gotten into the DeepMind science team. They were just finishing up work on AlphaFold 1, which already was pivotal work, really, but it hadn't solved the problem of protein structure prediction. And so I joined that team, and we were setting out to really, this time around with AlphaFold 2, solve it. And it was really this monumental task of how do we get there? How do we close this massive gap that existed? And we went at that for about a year and a half, 2 years of pure development. And I joined them as, an AI scientist and physicist with background in physics. And really, the team was comprised of a lot of people like me, either computer scientists or physicists. And together we came up with this novel algorithm, AlphaFold 2, that ended up, really being recognized as a solution to this problem in a competition called CASP. And so, famously by now, it's of course also been awarded the Nobel Prize for Chemistry last year, which went to Demis and John. And so really has had a tremendous impact on the field. After we've done that, I personally started work on protein design at DeepMind. So first that was just me, then we grew a team around it. I ended up co-leading that team at DeepMind and really became incredibly passionate about not just structure prediction, but really designing from scratch, generating protein with novel functions, and crucially having that connection going from bits to atoms, really making these things in the lab. And I found that intensely gratifying, and the impact that kind of technology obviously can have on the lives of everyone on this planet is so big that I wanted to really double down in this area. And eventually, I chose to found a company around that to really do that at speed and nimbly, and do that in a way that can benefit everyone in that ecosystem. And that's Latent Labs now. So I founded Latent Labs about 2 years ago, and really we're all about AI for drug design. We've been fortunate to assemble a world-class team. Others from DeepMind have joined. I'll stop here, and very happy to go into more depth in terms of our mission and how we're working and what we're working on. But really, that's Latent Labs, drug design, push-button drug design.
Thank you, Simon. Tremendous impact and tremendous science indeed. Now, the global pharma industry currently spends $300 billion annually on research and development, with a new therapeutic drug typically taking a decade to develop. With a shift towards a more personalized and democratic era of medicine, where treatments for rare cancers or debilitating genetic disorders are no longer a matter of luck, but of design, this can change. Research and development time and cost can be drastically cut, and patients able to access life-saving drugs faster, even in conditions previously deemed untreatable. Now, with this hope very much becoming a reality, Simon, I was wondering if you could, first of all, set the scene for listeners now on the AI protein design field, where it began, how it's evolved, and then describe for us where Latent Labs comes into all of this.
Yeah, absolutely. That sounds great. So I think ever since, we were starting to make drugs that went beyond sort of sheer serendipity, like discovering active compounds in nature, we sort of made mutations, we made changes to protein sequences in sort of a semi-rational way, and trying to actually influence potency, activity, all these properties that we're after. Initially, the field really worked on physics-based approaches. So that's actually one area that was, among others, pioneered by David Baker, who, of course, also was a co-recipient of the Nobel Prize alongside Demis and John, who sort of pioneered this idea of, yeah, trying to encapsulate the underlying biophysics of these molecules with functionals. And that, started to have, I think, good traction in the perhaps early 2000s, is fair to say, when the first de novo designed proteins were made. I think it's equally fair to say that while these approaches, were groundbreaking, they did not sort of generalize very well. So while people were able to make certain small proteins of certain folds, it wouldn't work on other problems. It wouldn't sort of generally say solve protein binding or enzymatic function. So it was quite a step, but at the same time still not a universal tool, and where you'd really want that. And that's equally, I think, showcased by what was possible at the time in terms of protein structure prediction. So going from sequence to structure, which is a fundamental part of understanding protein function, just wasn't solved until AlphaFold 2 came around. And so I think that's interesting to note, as we went from physics-based functional approaches to then machine learned approaches, with AlphaFold 2 perhaps as a catalytic moment where that really showed what was possible, how to apply machine learning in that space. And after sort of AlphaFold 2 came around, which perhaps you can conceptualize as a computational microscope, it allows you to visualize proteins found in nature. Then started the era, I would argue, of generative protein design and biology. And that's really what Latent Labs is contributing to, and pushing the envelope of. So really going beyond what nature gives us and controlling things like binding specificity, binding affinity, but then also, and that's perhaps where the state of the art in the field is, not just binding, but generating molecules, compounds that have desirable drug-like properties. And I think really that's maybe the bleeding edge frontier right now, to also have that co-designed. And that's something that we're really passionate about. We want to come to push-button drug design. So as opposed to, say, quote unquote, only push-button binder design, we want them to be, highly potent, selective, soluble, stable, and down the line, maybe one of the holy grail type properties is immunogenicity. Also have that mastered. I think that would be an intense milestone that would really unlock so much therapeutic potential. And as you mentioned earlier, I think that will also really give rise to personalized medicines once we, maybe can go much faster through clinical trials, because the compounds are better to start with.
Thank you, Simon. So within this generative landscape that we're currently in then, and going towards this push button drug design you mentioned, what about Latent X? I believe this platform has outperformed both DeepMind and isomorphic labs models in head-to-head testing.
That's right. Yeah, we launched our first frontier model called, as you said, Latent X earlier this summer, so just about 2 months ago. It's an all atom model. It's an innovative new type of model. The X in the name stands for all different binder types. So it's really generalizing across different modalities. So we've tested that on peptides, macrocycles, and mini binders. And it is, in head-to-head comparisons in the lab, achieving higher success rates and often better success rates compared to these competitor models that you mentioned. And why that's significant is it's really marking a shift from going from random discovery, if you will, from things like phage display, biopanning, or immunization campaigns. Basically, technologies where you have to screen really large libraries to arrive at it. This is now allowing us to test only very few and do that with good success rates and better affinities. We've actually since tested this technology more. We are competitive with the best macrocycle hit finding libraries out there. People that typically screen 10 to the 12, so that's 1 trillion molecules to find hits of the quality that our model generates within, say, 10 generations. So that's really a fundamental shift. And that's sort of the speed and the efficiency gains that we're seeing across the board. And as part of our launch, Latent X has actually been made available in a web platform and a API. And this is really kind of similar to, say, a chat GPT like interface. People, users, and customers from biotech and pharma can make designs literally at the push of the button. So you don't have to have AI backgrounds. You don't have to set up AI infrastructure. It's there for you to make designs immediately. And this has seen a really great uptick. It's being tested in tens of labs that we know of. There's good successes coming back. So it is really usable in the hands of our users. And we think that that really contributes to the sort of accessibility of tools like ours, which is, as you said, state of the art.
And I believe there have also been new proteins generated with latent X, haven't there?
Yeah, that's right. So we have made from scratch what people call de novo design, of course, macrocycles. We've made mini binders, and we're now actually pushing on further modalities of the likes of nanobodies and scFvs, and this is all looking incredibly exciting. So really, we can make biology programmable in that sense. We can set out and pick a particular modality that befits, say, a drug discovery program, and then make binders that are specific to the targets that we're interested in or that our customers are interested in. And what's exciting is that it very much looks like it's not just a speed advantage, but also unlocks desired design criteria that are really hard to achieve in the lab. To give an example, for instance, it's crucial, of course, that these molecules bind highly specifically, so they don't bind a similar but off-target target. And that's something that we can weave into the design process. And equally, there's difficult targets that have traditionally been deemed undruggable. Things like ion channels and the like, where we actually see good traction with these computational models. In part, that's because lab limitations such as the solubility of a membrane protein, they don't make a difference to our generative algorithms. So really, it's not just speed; it's also unlocking new targets, new design criteria that are otherwise really difficult to get a hold of with lab-based discovery.
As you say, it's exciting indeed. And the purpose of this, of course, is to democratize research. So I was wondering if you could explain why this intention is so very important now and in the future. And I want to add on another question as to what will set Latent Labs apart from, for example, open AI in this intention respect.
Yes, that's a fantastic question. So, as I mentioned, LatentX, our frontier model released this summer, has been made available with wide access in our web UI, available under platform.latentlabs.com. And really, users can make hundreds of designs for free every day. So we have published this in a free tier, and we think really this goes beyond, let's say, open sourcing the model. It's actually not open sourcing it, but it's making it available. And I think that the critical difference is that as opposed to making it available in some code repository, which will of course help computational scientists, the real sort of impact will be had in the lab when biologists start using it, when lab scientists start using it, when R&D drug hunters start using it. And for them, we want to lower the hurdles. And only then is it sort of going to really unfold the impact that it can have. So we really focused on that part. So making an intuitive interface, making it truly push button, giving interpretable confidence metrics that people can correlate with lab success, and we have shown that in our own research and published that, of course. And so it's about making it directly accessible where we think it will have the most impact, and that's sort of a philosophy we continue to follow. And I think it's sort of a win-win situation in the sense that users and customers have access to the technology in this very low lift way, while at the same time, we get to understand what the form factors of such a platform that really matter, what are the features that people want, and so that helps us improve our commercial offerings as well. So we think it's win-win, and yeah, that's how we're pursuing this. And then, as you mentioned, OpenAI, I think they're obviously an amazing organization at building general purpose language models, and that is speeding up and making more efficient a lot of work streams out of hard science. And I think that's where maybe the difference lies right now. We really focus on the hard science. We make the stuff work in the lab. At Latent Labs, we have our own wet lab, so we're able to directly validate at speed and scale what we're building, which gives trust to those who use our models. And equally, we're able to collect data that we can then use to improve our models. And so we're really focusing on the hard science. And I think that's the key difference where, that gives us an incredible edge when it comes to the applications that our customers want to pursue.
Thank you, Simon. So, lowering the hurdles to hard science with trust. I like this. So, I have a final question. If it all goes to plan, what does the future horizon look like in, say, 5 to 10 years from now?
That's a question I'm particularly excited about. So really, for us, it's about making good on that vision of going from biology being an observational science to making it a true engineering discipline. And what we mean by that is really, controlling as much of that hard science as possible computationally, so that the let's say 3 to 5 years that it takes to develop clinical candidates today actually is shrunk down to perhaps of the order of months or even faster, and at the same time enabling other undruggable biology so far. And so in the world that we foresee in 5 years, we will be able to get to the clinic much faster, and we will be able to do that at an order to 2 order to 3 orders magnitude more in scale. So we'll have much more shots on goal, and each shot will be much better. And we want to do that in partnership with, pharma and biotech. So as opposed to doing that in-house, we're about enabling this development at scale. And so I think that's where the power here will lie. And so when that comes to pass, well, that's in 5 or in 10 years, I think we'll be living in a world of essentially more abundance when it comes to drugs and what we can drug today. And I think it's a really optimistic and positive vision. That's a world that I would love us to live in, where almost all drugs will be curable. We'll have vaccines against many of them. We will have instant antibodies when pandemics hit again. Knock on wood, that doesn't happen, but I think we will all round be in control of our health.
Well, let's see what unfolds. It's been an absolute pleasure speaking with you today, Simon. Thank you for your time.
Thank you very much, Nicole.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.